Oh, okay. Slow to. Okay. Hi, I'm Jack Kosmicki. I'm a graduate student in Mark Daly's lab, and my time has been spent focusing on exome sequencing of our individuals with autism. So autism spectrum disorder, or ASD, is defined by the DSM as impairments in two core domains, first being communication and social interaction. The second is restricted interests and repetitive behaviors. It has a prevalence in the US of about 1 to 2%, and has a severe male bias in which there are 4 males with ASD compared for every 1 female with ASD. In terms of understanding the genetic etiology of ASD, most of the success, basically almost all of the success, has actually come from looking for de novo variants, and those are variants that are not inherited by the parents. They are inherited from the parents. They arose spontaneously in either the egg or the sperm. And so in this slide, basically all of the signal from de novo variants has come from protein truncating variants, or just also known as loss of function, or LOF, and missense variants. And so protein truncating variants are those that are expected to truncate the protein, so you get half of a protein or 3/4 or whatever, and most likely these proteins will go through nonsense mediated decay. And then missense variants are those that change the amino acid and may or may not have a effect on the protein. But what we've seen across these de novo studies is that there's a higher rate of de novo protein truncating variants and de novo missense variants that are observed in individuals with autism than what would be expected with that sample size. You can see that in that bottom corner or bottom box. However, for unaffected siblings, we see the rate of these de novo variants being what we would expect. Beyond just observing an excess of de novo PTV and de novo missense variants, there have been a number of other hallmarks of these consistent findings in these de novo studies. The first being that the rate of de novo missense and de novo protein truncating variants is higher in probands than their unaffected siblings. The second being that the rate of de novo protein truncating variants and de novo missense variants is higher in females with autism than their male counterparts. And this is interesting because remember that for every 1 female with autism, there's 4 males. So females seem to have more de novo protein truncating missense variants than their male counterparts. And lastly, that the rate of de novo protein truncating variants is higher in individuals with autism and intellectual disability than those with just autism. And so over the past 5 years, we now have a large number of de novo variants. So we now have de novo variants from close to 4,000 individuals with autism and a little more than 2,000 of their unaffected siblings. And so with all this data, we can now start to ask some questions, specifically those regarding some of the implicit assumptions that have been made about de novo variants in general. And there have been 3 main assumptions throughout these studies. The first being that mutation is uniform across the genome, so each site is equally likely to have a de novo variant. The second being that de novo variants are going to be rare and highly penetrant. And lastly, that it would be very unlikely to observe the same de novo variant present in multiple unaffected individuals. And so we sought to start to delve into some of these assumptions. We began by asking a very simple question. How often do we see a variant that is de novo in one family that's also present in other individuals in the population? And so here we have your standard trio. You have the mother, the father, and then a child. And here the child has a de novo variant at some site in the genome. And so we asked, how often do we observe that same site variant in the general population? And so, of course, we need a set of individuals to represent our general population. So we turned to the Exome Aggregation Consortium, or EXAC, which was put together by Daniel MacArthur and Michael Leck from Massachusetts General Hospital. And this is the largest reference exome database. So it's close to 61,000 individuals without autism from a variety of ancestries, and this is almost an order of magnitude larger than the exome sequencing project, which was the largest reference exome database before. And while this isn't a perfect representation of the general population, it's certainly better than what we've previously had. So when we actually go in and look to see whether any of the de novo variants that were present in our autism cohort are also observed in EXAC, we see remarkably that about 1 in 3 of these de novo variants are at one of these sites in EXAC that's variable. So there are people in EXAC with one of these variants. So we have a de novo variant that was thought to be perhaps causing autism in an individual, and we also have that same variant existing in an individual in EXAC. So this was a rather surprising finding. The first one we checked is whether these de novo variants are more likely to occur at certain sites than others. And in fact, as you recall, I mentioned that we made this assumption that the mutation rate is uniform across the genome. Well, we know it's definitely not. Especially at methylated CpG sites, have a mutation rate that's an order of magnitude larger than the overall mutation rate. And so we see that 71% of these de novo variants that are present in EXAC occur at these CpG sites, versus about 29% of the de novo variants that we didn't see in EXAC are at a CpG site. So this is like highly significant and makes sense from a biological point of view. Because if you're going to choose a site in the genome to mutate more than once, you're probably going to choose a site in the genome that's more mutable, so CpG sites.
Just a quick interruption.
Yeah.
I just heard that the people see the presenter mode.
Oh, actually are not seeing the full.
Yeah. So
okay.
Maybe I just can you maybe change this here?
How do I see?
Just mirroring, I think, would be.
Let's see. Sorry about this. Let's see.
Well, in PowerPoint.
Is it like perfect displays, and then you just choose
arrangement? I think you can do mirroring. That's already mirroring. So then in presentation mode, so it'll be back to PowerPoint.
Okay.
There. Okay. Slideshow. Or view, maybe. Or we can do this more easy if we just get rid of this. So no, there should not be an external. Okay. Yes.
Okay.
You can just move this.
Oh shoot.
Yeah. Left. Yeah. Let's put it somewhere. Okay. Let's go ahead.
Okay. So sorry about that. This was everything. Okay. So we see that. Excuse me.
You have to share it again. Sorry for the interruption. So PowerPoint, share screen should work. No?
Yeah. No. Okay. Sorry about that. So we've observed that basically 32% of our de novo point mutations are at one of these sites that are variable in EXAC. And so then this begs the question as to whether the de novo variants that are present in EXAC are any different from those de novo variants that we have yet to see in EXAC. So we started with looking at the rate of de novo synonymous variants, and these are the variants that don't actually change the amino acid and thus don't change the protein. And so in purple, we have the rate of de novo synonymous variants in individuals with autism, and then in green is the rate in their unaffected siblings. And then in blue on the far right is just the rate of de novo synonymous variants in individuals with autism and developmental delay. And so across all three groups, there's no difference in the rate of de novo synonymous variants, which is what we would expect, given that these variants are we expect them to be neutral as a class. And so when we look at just those de novo synonymous variants that are present in EXAC, we again don't see any difference, nor do we see any difference in the de novo synonymous variants that are absent from EXAC. So regardless of how you slice your set of de novo synonymous variants, as a class, we're not seeing any difference between individuals with autism nor their unaffected siblings. On the other hand, if we move to our de novo protein truncating variants, and these are the variants that have shown the largest effect size in autism, we see that here, if we look at all de novo protein truncating variants, they're like 1.6 times more in individuals with autism compared to unaffected siblings, and 2.2 times, there's a 2.2 fold excess in individuals with intellectual disability and developmental delay. Now, when we just look at those de novo protein truncating variants that are present in EXAC, now we don't see any signal. There's no signal whatsoever. And in fact, there's a higher rate of de novo protein truncating variants in unaffected siblings than individuals with autism or intellectual disability. So mathematically, when you remove the de novo protein truncating variants that are in EXAC, you're left with a larger excess in individuals with autism and intellectual disability and developmental delay. So we've gone from, a 1.6-fold excess to now a 1.8-fold excess for autism, and 2.2 to 2.6. So we're seeing a larger excess than the full data set when we remove those de novo protein truncating variants that are present in EXAC. So this lends us to, this basically shows that these de novo variants that are present in EXAC are less likely to confer risk to the phenotype, in this case autism, than those variants that are not seen in EXAC. And now we can go forward and look at some gene level analyses. So instead of looking at specifically whether a variant is in or not in EXAC, now we can ask, are there genes that are now overrepresented have a lot more de novo variants than we would expect? So here we're asking how many de novo variants absent in EXAC reside in these genes that are predicted to be loss of function intolerant. And so we used a recently developed metric by Caitlin Samoa, a postdoc in our lab, called PLI, which is just the probability of loss of function intolerance. And genes with a high PLI score are more likely to act similar to haploinsufficient genes, in which so haploinsufficient genes are genes in which if you lose one copy of the gene, then the gene doesn't work. So if we split our de novo protein truncating variants that are not in EXAC between genes with a high PLI score, so genes that are more likely to act in a haploinsufficient manner, from the remaining de novo protein truncating variants, we see a higher excess. We basically see that all the signal is in these de novo protein truncating variants that are absent in EXAC and in one of these likely haploinsufficient genes. So if we look at where we've gone and where we are now, we started with a 1.6-fold excess in autism. After we removed all the de novo protein truncating variants that are in EXAC and all the de novo protein truncating variants that were not in EXAC but in one of these genes that's unlikely to be haploinsufficient, we arrived at now a 3-fold excess over unaffected siblings. And this excess is even larger with intellectual disability and developmental delay, close to a 7-fold excess. So now we can ask about what about those other hallmarks of these de novo studies? Do we see a larger rate of protein truncating variants that are absent in EXAC and in PLI genes in females than males, and do we see a skew in the IQ distribution? So if we look at the female to male ratio, that in this box, 6% of the individuals with autism in our cohort have a de novo protein truncating variant that is not in EXAC and in one of these high PLI genes. And there's 2 females for every 1 male. So this is far more females that have one of these de novo protein truncating variants than their male counterparts. And if we look at all the other individuals that have one of these other de novo protein truncating variants, which are 7%, and then all the individuals that don't have a de novo protein truncating variant, here the male to female ratio is about 1, or is a little less than 1, so it doesn't, and then their IQ is also generally about the same. So what's interesting is that not only do females have a higher rate of these de novo protein truncating variants and likely haploinsufficient genes than their male counterparts, but also if you are an individual with autism with one of these other de novo protein truncating variants, or you don't have any de novo protein truncating variants, you look pretty much the same. You have about the same IQ and the same male to female ratio, which is pretty interesting. Then if we look at the skew in the IQ spectrum, that curve, the curve here, is the curve for de novo protein truncating variants absent from EXAC. And here we see that skew in the IQ distribution. So you see there's a higher rate of de novo protein truncating variants absent from EXAC in individuals with low IQ than those with a high IQ. And if we look at all those other de novo, all the other de novo protein truncating variants, there's no skew in the distribution. It's quite uniform across IQ. So when we remove a third of our data set, not only do we get like a larger, we're finding a larger excess in individuals with autism, but we're also seeing the same male to female ratio, and we're seeing the same IQ skew in the IQ distribution. So we're not like losing any signal that we saw in the full data set. So the other thing is we know that, you know, de novo variants contribute a small portion to liability of ASD, and we were curious if we perform the same analysis, do we see an excess in inherited protein truncating variants? And so on the top here is just the rate of de novo protein truncating variants that are not in EXAC, one of these likely haploinsufficient genes. And here on the bottom is the number of rare inherited protein truncating variants, or rare protein truncating variants that were not inherited. And so we see for the rare protein truncating variants that were absent in EXAC and in one of these likely haploinsufficient genes, we see a small excess. So they're more likely to be transmitted to an individual with autism than not be inherited. And then for all the other rare protein truncating variants, of which there's about 12,000 of these that are both inherited and not inherited, there's no difference in whether it's these variants are not more likely, are equally likely to be inherited. You can see just by the raw numbers that there's about like 670 of these of these inherited protein truncating variants that are inherited and show signal as compared to the overall, we're looking at like 12,000 of them in our cohort. And so you can see there's just a very small slice of the pie that's actually showing any kind of signal. It's quite small compared to no variants. And lastly, just when we look at our genes, the genes that have been implicated in contributing to autism, and we look at just the raw numbers, we see that very few of these genes actually show any signal, or there's a few variants that are inherited, and there's a few variants in cases, but basically we still get the same list of genes that are contributing to autism as we originally saw. So we're not changing, we're not losing signal, basically. And so in summary, basically all of the excess de novo and inherited PTV signal resides in these variants that are absent from EXAC and in genes that are intolerant of truncating mutations, genes with a high PLI score. And lastly, we see that basically all individuals with autism have about 3.5 protein truncating variants, both de novo or inherited, but only 4% of these protein truncating variants actually occur in one of these genes that's intolerant of such variants. And these the variants that appear to be contributing to risk. And with that, I'd just like to go to my acknowledgements, and I'd like to thank my PI, Mark Daly, and a bunch of other people that have helped me along the way with this analysis. If you have any questions, my email is like on the bottom.